All right, well, I hope you guys are doing well. This is our last chapter for this class, and it's chapter 26. We're going to lecture over it in 2 parts. And I hope you guys are ready and excited to be at the end of the semester. I know it's been an unusual semester. We are nearly there. So we're going to do about half of this 30-slide lecture for today, and then we'll complete the other half on Wednesday. So here's an example of where genetics can be seen in our physical world. So whether or not your earlobe is free or attached depends on the genes you inherited. So you guys did a genetics lab this last week, which you may have hopefully looked at people around you, like your family, your small group, family, to see how these genetics might play out in your family. So for example, on the left, this is a free earlobe, meaning it's not attached there at the bottom. And then on the right, there is a piece of skin attaching the bottom of the lobe, which would be an attached earlobe. So what is a gene? Well, it's a portion of DNA that determines a specific characteristic. The locus is the site on the chromosome where a particular gene is located. During meiosis, diploid, which are the 2N cells, are converted into haploid, which is half N cells. The diploid goes through meiosis, becomes a haploid gamete. The diploid organisms, you get 2 sets of chromosomes, 1 for each parent, so we are diploid. And then there are 2 genes for each characteristic, 1 gene from each parent. So meaning one parent might have a free earlobe and the other parent might have an attached earlobe, and then what you inherit is a Mendelian genetics, which we're going to talk about, which is probability, a lot of probability, but not always. Okay, so this is how these genes are passed along in diploid organisms through meiosis. So alleles are alternative forms of a gene within a population. So for example, this is free and attached earlobes. You could see that there's, all in all, it's probably a 50-50 would be my guess. Half of us have attached and half of us have free earlobes. They're located on the same pair of homologous chromosomes at the same locus, meaning your earlobes that you have that are free are the same gene on the same chromosome on the same locus at the same locus as somebody else who has a free earlobes. It doesn't come from different places. It's always the same specific chromosome, the same specific site on the chromosome. Genome is the set of all the genes necessary to specify an organism's complete list of characteristics. So you, from the tip of your head to the tip of your toes, everything that goes into expressing everything about you physically comes from the information in your genome. And so any sort of abnormality in your genome could result in a phenotypic expression that is different. So say like 6 toes or an incompletely formed arm. These are examples where something happened within the genome, the development, where it didn't go quite right, but it comes from the instructions, which are your genome. So genotype, we talked about genotype as in the lab as being all the genes present in an organism, consists of your DNA code, but you cannot see genotype. We can take a sample of blood and send it off to a lab and determine what genes you might have. If you have certain genes, like I have high platelets, and so one of the things my doctor did was look for genes which might suggest something bad going on in the background causing the high platelets, and so that's my genotype. We can't see if I have these genes that might be causing high platelets, but we can test for them because we know where to find them. Now, phenotype is all the observable characteristics of an organism. So hair color, eye color, ears, curly hair, straight hair, dimples, no dimples, widow's peak, no widow's peak. And these are determined by the ways in which the combinations of alleles express themselves. So most of your things like eye color and hair color, well, I don't know about hair color, but specifically eye color, it comes from more than one allele. So it's a combination of things, not just one. Now, your gene expression is the degree to which a gene goes through protein synthesis to show itself as a physical feature of the organism. So just because you have a gene doesn't mean that it's going to be expressed. Only the genes that go through gene expression are going to be physically expressed as hair color, eye color, and things like that. If you get a recessive gene from a parent that isn't, say they have curly hair and you have that recessive gene, but the dominant gene is straight hair, only the straight hair gene is going to be expressed. The curly hair gene is not going to be expressed, even though it is part of your genotype. It is not part of your phenotype. So the model of inheritance relies on two different types of allele. The dominant allele, which masks other alleles for the trait, meaning it's going to be dominant, and designated by a capital letter. Okay, so we show dominance by this capital letter. The recessive allele, on the other hand, is overshadowed if there is a dominant allele around. And we're going to designate that by a lowercase letter. So if you have two recessive alleles, one from mom and one from dad, and they're both lowercase letters, then you have the recessive gene. But if one parent has a dominant allele, then that's going to dominate the recessive one. When both alleles are the same, meaning both uppercase, both lowercase, both dominant, or both recessive, we're going to call that homozygous. Homo usually always means same. Zygos in this case is referring to your alleles. If you have two different alleles, which would be a dominant and a recessive, that's the only different you can have. This is going to be a heterozygous combination because they are different. So the genotype for two alleles for free lobe, it's the two dominant EE, so it takes two dominant alleles. Two alleles to have an attached earlobe would be lowercase e, and so these are when your earlobes are going to be attached, and that's the recessive. Now, and those are the homozygous options. The heterozygous option is one capital E and one lowercase e, but because the dominant trait is the free earlobes, it means that if you inherit a capital E and a lowercase e, you're still going to have free earlobes. Physically, we're going to see that you have free earlobes, but your genotype is actually capital E, lowercase e. So you can see here that we can physically see the phenotype, but I wouldn't be able to for certain say what your genotype would be. It could be two capital E's, or it could be a capital E and a lowercase e. We only know for certain when you get the recessive trait that you have both the recessive alleles. So on the bottom here, there's an important part in red. It says 3 genotypes, but only 2 phenotypes. Okay. So Gregor Mendel, he is, and it doesn't say here, I'm surprised, but when I took genetics, I learned him to be the father of all genetics. And I feel like that's probably a little bit of a disservice to people who also specialized in genetics and made contributions. But Gregor Mendel is known for his studies with pea plants. So he's an Austrian monk who performed experiments, whether or not he called them experiments at the time he was doing that showed the inheritance patterns of certain characteristics of pea plants. But the good thing he did was he had a controlled experiment. And what does that mean? Well, a controlled experiment is where we're only varying one trait at a time. It means we're not going to vary the amount of sunlight and the amount of water and change the soil and then say, well, the reason why this plant grew better was because it had more sunlight. Well, you don't know that. It could have been the soil. It could have been the water. You can only change one thing at a time to know for certain what's the driving force behind the change that's observed. So from his controlled experiments, Gregor Mendel determined he could conclude which traits were dominant and which traits were recessive, which is a very interesting thing to do with pea plants. Especially when he was doing this, we didn't know anything about the DNA. We had not sequenced the DNA, and we knew very little about genotype and phenotype. And so what he did very early on was establish some principles that are still true today. So, for example, some of the characteristics that he studied were plant height, pod shape, pod color, seed surface, seed color, and flower color. He determined which ones were dominant, which ones were recessive, and then was able to use that to do very specific breeding of these pea plants. So Mendel's laws of heredity, there's 3 that we're going to talk here. The first is law of dominance. When 2 different alleles for a given trait are present, the dominant allele is expressed and the recessive allele will be overshadowed. Okay, so that's the law of dominance. If you have a dominant allele present, it's going to overshadow the recessive one. The law of segregation is when gametes are formed by a diploid organism, the alleles that control a trait separate from one another into two different gametes. So if you think about it, if your parent had a capital E, lowercase e for earlobes, and that chromosome is going to break into two different sperm or two different eggs, meaning one of the dominant E's is going to go to one and a lowercase e is going to go to the other. And which one gets fertilized or which one does the fertilization is really outside of our control. So the law of segregation states that because the diploid organism breaks into two separate organisms and only one of them goes on, in our case for humans, to be the offspring, we don't necessarily control which one gets fertilized. In plants, it's kind of similar. But what that means is once we look at the offspring, we can go back and predict what we think the genotype of the parent plant might be. The law of independent assortment, members of one pair of alleles separate from each other independently of the members of any other pair of alleles, meaning that they are not going to be, what? That's mine. That's my Zyrtec.
Yeah,
it was sitting on the counter because I'm going to take it at lunch. Thank you. The members of one pair of alleles separate from each other independently. So there's no, say that we have a gene that are like your eye color comes from more than one gene. So it's a poly. Many. And so say that it takes, 3 of them being dominant and 2 being recessive for you to have brown hair, and you don't necessarily, just because you get the dominant allele in one of the sets doesn't mean you're going to get the dominant allele in all of them. And that's an important protection against mutations and against passing down genetic defects and things like that, is that it is kind of like throwing all the cards up in the air and just picking 26. Because if you have 52 cards and you throw them all up in the air and you only get to pick up 26, the first card I pick up has no bearing on the 9th card I pick up. And that card has no bearing on the 17th card I pick up. They have no control over any of the things. It's an independent assortment, and you get what you get. Okay? So single factor crosses are going to be genetic crosses in which a single characteristic is followed from one generation to the next. So here's the problem. If 2 parents are heterozygous with respect to the allele for Tourette's syndrome, what is the probability that they can have a child without Tourette's syndrome? So both parents are heterozygous, meaning they're going to be the middle column right here where they have the capital T, lowercase t. So that's what you have to know. Capital T means you're going to have Tourette's. Lowercase t means that you're normal. So we're going to assign a symbol for each allele, usually capital and lowercase letter, based on what's dominant and what's recessive. And it's single factor because there's only one set of genes that we're looking at, okay? So now we're going to set up what's called a Punnett square. And so if you look up in the red, the first column, the first row with the red words in it, that's your female genotype. And so right underneath that, in the second column, you have your it doesn't matter which one's which because they're both heterozygous. So you're going to have your capital T, lowercase t. So in the second row across and the second column down, the capital T and lowercase t, those are your parental alleles coming to the table, okay? Now remember that these are going to separate. The law of segregation says they are going to both sets are going to separate into 4 independent alleles. And how they come back together, we don't really have the exact control on, but we can predict with statistics what we're likely to see. So then what we're going to do is we're going to start matching up the columns. This is like that really weird math that they're doing nowadays where you set up this rubric. So we're going to take the capital T and the capital T, and we're going to pair them up as they show up in like a crossword puzzle. Then the capital T is going to pair with the lowercase t in the final column. I wish I could write on this. I wonder if I can. Nope, my finger won't work. Okay. So the capital T and the lowercase t are going to match up in the third row, fourth column. And then in the bottom row, the lowercase t is going to match up with the capital T in the third column and the lowercase t in the fourth, in the final box. And so there is a typo. In the bottom right-hand corner, this should be lowercase t, lowercase t. Okay, in the bottom right-hand corner, because the lowercase t and the lowercase t are coming together. And so it tells you that all possible are produced using the Punnett square, which is what this is, determine the gene combinations, and now we have to go back and look at here. If the genotype is either capital T, or capital T, lowercase t, the baby or the offspring is going to have Tourette's. The only time the baby will not have Tourette's is if it has both alleles of a lowercase t, lowercase t. And so of the 4 options we have, 3 of them are going to result in the baby having Tourette's, which means 3 out of 4 is 75% chance. There is only a 1 out of 4 chance, 25% chance that the offspring will not have Tourette's. The other way of thinking about this is if the parents have 4 children, 3 are likely to have Tourette's and 1 is likely to not. Okay, so it's either 75% chance you will, or that 75% of their offspring will. Okay? Now, it obviously gets more complex than that. The phenotypes are usually determined by the allele, the alternative ways in which they react, as well as the environmental influences in their expression. There are 6 recognized patterns in inheritance that we're going to talk about. X-linked characteristics, codominance, incomplete dominance, multiple alleles, polygenic inheritance, and pleiotropy. So, for example, here, this horse right here has like quote-unquote natural highlights. It is not completely all the way around a red horse or brown horse. My husband would know a bay. I think they call these bays. It's not completely bay. It has white flecks of hair in there, which suggests there might be something else going on with the inheritance of the bay gene. So X-linked genes are going to be alleles when they are located on the same chromosome that tend to be inherited together. They are linked, meaning if you get one, you are likely to get the other. The closer they are, the more likely they are to be linked. So an example here is the sex chromosomes, the X and the Y. Females are XX, males are XY. The Y is much shorter than the X chromosome. It does not have many of the alleles that are found on the comparable portion of the X chromosome. So, if you are a female, you have more alleles on your 23rd chromosome than a male. In males, the presence of a single allele on the X chromosome will be expressed no matter if it is dominant or recessive, meaning there's not as many options. Remember, because we talked about that on each allele there's going to be one inherited from mom and one inherited from dad, meaning if you have an X and a Y, you're missing a portion of your chromosome in that 23rd set, which means you're not going to have 2 different genotypic expressions to pull from. You're only going to get what's expressed on the X. So X-linked genes are found together on X chromosomes, more than 100 genes, color deficiency, hemophilia, muscular dystrophy, brown teeth. So male pattern baldness, maybe it's called female pattern baldness. I don't know. But baldness is a really interesting thing because you actually inherit that from your mother's grandmother. And that has something to do with the fact that your X chromosome, the 23rd one that's coming from mom, well, it's it could have the baldness on there. And so you're actually getting that from your mom and not from your dad, which is strange because you tend to look at your dad and say, well, if he's balding, I'm going to bald. But not necessarily. The other thing is like hemophilia, muscular dystrophy. Hemophilia is more common in males. And that could be because of the fact that they're linked to the X gene, and with the Y gene, there's nothing counteracting that recessive gene. Color deficiency as well. Men tend to be more likely to be colorblind than women. Y-linked genes, the SRY, controls the differentiation of the embryonic gonad to a testes. We're not going to worry about that. Do not worry about that last statement at all, because we didn't talk about that specifically when we did human reproduction. Okay? So you just need to know that some of the things that males are more susceptible to, color deficiency, hemophilia, muscular dystrophy, brown teeth, come from the fact that they lack those extra alleles on their 23rd chromosome because it's a Y and it's shorter. Codominance is when neither allele is dominant or recessive. The phenotype of each allele is expressed in a heterozygous organism. So the phenotype of an individual can be very different from either homozygote parent. In this case, all uppercase letters and superscripts denote the allele. So, for example, the color coding of horses and cattle. To have a true red coat, it'll be CR, CR, to show that it's a true red coat. True white coat would be CW, CW for white horses coloring. Now, the roan is the color we saw before, which I probably should have known that, is where you have the red and the white hairs. And so you're going to have both CR and CY, because neither one of those is dominant. The horse isn't going to be red or white. The horse is going to be both red and white. Okay? So that's codominance. They're both going to be expressed. So, for example, in these flowers, these are snapdragons. The parental snapdragons are red and white, respectively, in the images. But because the red and white color is codominant, the offspring plants are going to be pink, because red and white make pink. Now, incomplete dominance is where the alleles lack dominant relationships. So heterozygous offspring have intermediate phenotypes not distinct from either homozygote parent. So snapdragons in the previous one, that is incomplete dominance. Two alleles for the parents, white and red. Neither one of them are recessive. So because neither one of them are recessive, you're going to have an incomplete dominance where they kind of both are going to show. The allele for the white petal is FW, for F for W for white, and then the allele for the red petal is FR. Both red and white flowers alleles partially express themselves, two different alleles but three different phenotypes. So the flower could be white, red, pink. All right, multiple alleles. So multiple alleles where all forms of the same gene that control a trait, more than two different alleles for a single trait. One organism can have a maximum of two alleles. So for example, blood type is where you're going to have three alleles, A, B, and O. A and B show codominance. A and B are dominant over O, so meaning if you inherit both A and B, your blood type will be AB. If you inherit an A and an O, your blood type is A. If it's a B and an O, it's B. The only time you will be type O is if you inherit both O and O from your parents. We're not going to talk about the antigens, so don't worry about the first table. And then for the genotypes on your table on the right, you do need to know those. And so I'm going to go through them so that you can fill them in, because I'm not sure why they're not showing up on the table. But to have blood type A, your options are AA, AO. For blood type B, it's BB or BO. For blood type A, it's AB. And for blood type O, it must be OO. And they're all capitals because they're all the alleles. There's no recessive one there per se. Okay? So again, it's AA, AO, BB, BO, AB, and OO. Okay, so make sure you know those. It'll show up as a multiple choice question on your test. Okay? All right, so let's do one more thing. We'll do polygenic inheritance, and then we'll stop here. We'll do two more. We'll get to pleiotropy. Yeah. Okay. To finish out this part. So polygenic inheritance, characteristics determined by interaction of genes at different loci. Number of different pairs of alleles determine characteristics. So in this case, your skin darkness or lightness is going to be polygenic. So it comes from more than one genotype. And so it's not a single trait. If we had a Punnett square before, the Punnett square was going to be only like T, capital T, lowercase t, capital T, lowercase t. But this time it might be capital T, lowercase t, A, and B. It's going to be a huge Punnett square that makes up your skin type, because we know that there's a variety of things that go into it, not just your skin color, but then you have skin tone, which could be olive, red, yellow, green. We have different combinations that contribute to your skin type. And so that's what polygenic inheritance is. And you can see here that there is a variety of shades of color. If you are a lady and you have had to buy foundation or concealer, any sort of makeup to match your skin tone, you know that there are a variety of shades. I worked at Walgreens in college and used to have to stock the makeup. And, you know, it's like 101 and 104 and 103. And you just really have to find the best shade, but you never really find the true shade, even when they tell you, this one's going to match your skin. It's like, yeah, but only for a week during the summer, or only for that week during the winter when I'm so pale. So it's because it's such a complicated formula. All right, so pleiotropy. This is where a single gene has multiple effects on a phenotype. So this is crazy, because you'll have one gene that causes multiple physical expressions. So for example, Marfan syndrome, this is where I think if you've ever seen images of Abraham Lincoln, they thought maybe he had Marfan syndrome. It's pretty commonly diagnosed, not pretty common, but it can be commonly diagnosed in basketball players, because they tend to be very tall, tend to have very long arms, very long legs, I would say very slight build. You can see here that they're wrapping their fingers around someone's wrist, very long hands, long fingers, very slender build. You can see right here on the right hand side. The other one is phenylketonuria, and this is the PKU. And so this is where you're going to have I was going to look it up, because I know when Anderson was a baby, they tested him for this. Yes, and so this is a defect that causes an amino acid called phenylalanine to build up in the body. And so the U.S. screens babies for this because the accumulation of the phenylalanine in your body can really devastate the body. But some of the things are brain damage, intellectual disabilities, behavioral symptoms, or seizures, and they want to know right away if a baby has it. And so normally, when he was in the hospital, they pricked his foot. It was the worst thing because his feet were so tiny, and he screamed bloody murder. And then they put it on this little card, and they send it off for testing. And so Marfan syndrome, there's really nothing that can be done to prevent it once you have it. But you just live a life with a little bit of modifications, because I think the biggest concern is overtaxing on your heart, and you might have weakened heart structures. I actually just watched an episode of Grey's where the guy, if you've ever seen it, season 2, like episode 6, where Christina and Dr. Burt go out to dinner for the first time outside the hospital, and then the patient goes into like cardiac arrest, and he has Marfan syndrome. And so that's so strange, because I just learned about that, just saw the show about that. So this is the pathway for the PKU. You do not need to worry about this slide at all. Okay, just know what pleiotropy is, and you'll be good. All right, so we're going to finish up the rest of the genetics on the next lecture, which should be a little bit shorter, so we made it a little bit further. And so make sure you turn in your notes at the end of this week, because we are getting so close to the end of the semester.